In this video, we will be covering 4 types of point mutations, including missense, silent, nonsense, and read-through mutations. Before we go into the types of point mutations, let's go over transition versus transversion mutations. Transition mutations occur when a purine mutates to another purine or pyrimidine mutates to another pyrimidine. For example, this occurs if an A or G mutates to a C, U, or T, or vice versa. Now let's go over transversions. Transversion mutations occur when a pyrimidine mutates to a purine or vice versa. So an example of this would be an A or G to a U, T, or C, or vice versa. Now let's start by looking at a normal DNA coding strand. Before we can begin translation, we must transcribe the DNA into mRNA. Since we are given the coding strand, the mRNA will be the same as the coding strand, except uracil will replace the thymines. Once this is complete, we can begin transcribing the mRNA into a protein. First, we must find the start codon AUG. Remember that you read mRNA from 5' to 3' and that each codon is only 3 letters long and they do not overlap. As we read codons, we can then begin identifying what amino acid they code for by looking at a coding dictionary. We continue down the strand until we reach a stop codon. In this case, it will be UGA. Once we know what this normal mRNA looks like, we can begin looking at some mutations. First, let's begin with a missense mutation. A missense mutation occurs when a single nucleotide is mutated into another. For this example, let's change the AAC to AUC. Since the A has been mutated, a new codon is presented which codes for a different amino acid. In this instance, an isoleucine would be inserted where an asparagine would have been. Since this mutation was from an A to a U, this is a transversion mutation. Next, let's look at a silent mutation. In a silent mutation, while a nucleotide has been mutated, the codon will still code for the same amino acid. For example, if we change UGC to UGU, the codon would still code for cysteine, thus not changing the amino acid sequence. Since this mutation changed from a C to a U, a transition mutation occurred. Next, let's look at a nonsense mutation. In a nonsense mutation, a codon that would originally code for an amino acid becomes a stop codon. For example, if in the codon UGC became UGA, this transversion mutation would result in the original cysteine codon now coding for a premature stop codon, thus ending the sequence early. Finally, let's look at a read-through mutation. This mutation occurs when a stop codon is mutated to a code for an amino acid, thus causing translation to occur past where it should have stopped. For example, if the UGA were mutated to AGA, then when the ribosome comes to this codon, instead of stopping, it would read it as an arginine, then proline, then glycine, instead of stopping as it should. This pyrimidine to purine change is a transversion. All references are found in the lecture slides and the book found in the description. Thank you.